Hi everyone, so today we'll be going over enzyme kinetics, which is the study of the reaction rates of enzyme catalyzed reactions. Now I've included two separate notes for this lecture because there's a bunch of equations. So you could either use the notes with the equations already written on them, or if you like to follow along with and write the equations for yourselves, I have included ones with no equations on it so you can fill them out for yourself. So we'll spend most of the lecture going over a bunch of different variables that are used to measure enzymatic activity. So we'll learn about things such as maximum velocity, the Michaelis constant, turnover number, and these may seem a bit daunting at first, but I promise if you just sit down, give them some thought, they're really not that complicated. So we'll end the discussion with a short talk about specifically enzymes that catalyze reactions with two different substrates, but that'll be pretty quick. So let's get started with a pretty important lecture, Lecture 8, Enzyme Kinetics. So today we're going to start talking about enzyme kinetics. So enzyme kinetics is the study of the rate or velocity of enzyme catalyzed reactions. So we'll have a lot of equations today, and so we'll go through those, but we'll go through pretty slowly so everybody can follow along. So if we think about a reaction of A goes to P, so A would be our substrate or our reactants, and then P would be the product. Now, you can measure the rate or velocity, which is signified by the letter V, of this reaction by two different ways. So one is you can measure the decrease in the concentration of A over time. So the decrease in the concentration of the substrate or reactant. So that would be written as the velocity, V, as a function of decreasing concentrations of A as a function of time. So this would be the decrease in this concentration of A over time. Or you could look at it as an increase of product concentration over time. So that would be written as V, or velocity, equals a function of the product increasing as a function of time. So the velocity of the reaction is directly proportional to the concentration of A or P, but let's keep it with A. So you could also more simply write this reaction as the velocity equals K to the concentration of A. And that K is going to be our rate constant. And for this type of reaction, the units of that rate constant are going to be per second. So second to the power of -1. So it'll be per second. So the units of A are going to be in molar because that's the unit of concentration. So this type of reaction would be a unimolecular reaction because we only have one substrate that gets converted into products. And the rate is going to be directly proportional to the reaction concentration. So that would be called a first order reaction. Now, a second order reaction would be a reaction that includes two reactants. So this is called a second order reaction. So there's a few different ways you can describe this. So you can do, A plus A is converted to product, which can be simplified to just 2A is converted to product. Or it could be written, if there's two different types of substrates, it could be A plus B is converted to product. So now the rate equations for these reactions would be a little bit different than what we saw for the first order reactions. It would be the velocity of the reaction equals the rate constant times the concentration of A squared, or the velocity of the reaction equals the rate constant times the concentration of A and times the concentration of B. So the units would be a little bit different in this one. So the units of the rate constant would be per molar per per second. So molar to the -1 times second to the -1. So this is how reactions be written in second order reactions, where the reaction rate or velocity depends on the concentration of two different substrates. So now that we've just looked at chemical reactions, now we can add in enzymes to make equations that account for enzyme catalyzed reactions. So an enzyme catalyzed reaction would be described by an enzyme. You have an enzyme plus a substrate, so S for substrate, and this is a reversible reaction where this will form an enzyme substrate complex as an intermediate. And that enzyme substrate complex can either revert back to enzyme plus substrate, or it can be converted in a reversible reaction into enzyme plus product. So you have the substrate is converted into a product by the enzyme, and there's this formation of an enzyme substrate complex. And the enzyme substrate complex can either convert back to enzyme plus substrate or convert to enzyme plus product. Now we can add a rate constant for each of these forward and reverse steps of the reaction. So if we have enzyme plus substrate forms the enzyme substrate complex, so that would be the intermediate of the reaction. And then that will form enzyme plus product. We can add rate constants for each of these steps. So the conversion of enzyme plus substrate into an enzyme substrate complex can be described by K1. And the conversion of the enzyme substrate complex to enzyme plus product would be K2. And then the reversible parts of that, so if enzyme plus product goes back to the enzyme substrate complex, that would be K -2. And then if the enzyme substrate complex returns back to enzyme plus substrate, then that's K to the -1. So these are the rate constants that we're going to use to describe each of these steps of this reaction of the conversion of a substrate to a product by an enzyme. Now, it's useful for reaction kinetics that we look at early time points in reactions, and I'll get to that a little bit more in a second. But at early time points before product accumulates, this equation can actually be simplified by ignoring the conversion of product back to substrate. So that would be the rate constant of K to the -2. So a better way to write this, if we're going to describe enzyme catalyzed reactions, would be enzyme plus substrate is converted reversibly to an enzyme substrate complex. We have K1 is the rate constant for the forward reaction. K to the -1 is the rate constant for the reverse reaction. And then the enzyme substrate complex, just one direction, would turn into enzyme plus product, and that would be described as K2. So this allows us to simplify the equation for an enzyme catalyzed reaction a little bit, which will make a little bit more sense here in a minute. So a common way to investigate enzyme kinetics is to measure the velocity or rate of a reaction as a function of increasing substrate concentration with a fixed amount of enzyme. So if you're going to investigate the rate of a particular enzyme, you do an experiment where you keep the enzyme concentration the same, and then you just add increasing amounts of substrates. And this gives us a lot of information on how quickly the enzyme works. So, another important feature is to look at the initial velocity of a reaction. So that's the rate of the enzyme conversion of substrate to product at the beginning of a reaction before product accumulates. So you can take that variable out of the equation. So when the product equals 0 and the time equals, basically 0, you can determine the initial reaction the initial reaction velocity at increasing substrate concentrations. So here on the left, we have a graph showing the product formation over time with increasing substrate concentrations. And so this initial velocity, or v sub 0, is going to describe, it's going to be the linear plot of that reaction rate at the very beginning of the reaction before product accumulates and when time is basically 0. So you can take those variables out of it. And when you end up plotting these initial velocities for enzymes, it gives you a hyperbolic curve like this, where it goes up very quickly and then it starts to plateau out. So here on the y-axis would be the initial reaction velocity, and at the bottom is that increasing substrate concentration. And then there's going to be a maximal velocity point where the enzyme is going to reach its basically maximal rate of it can't really convert the substrate into product any faster than it does. So when you're doing experiments like this with a fixed amount of enzyme and increasing substrate concentration, this allows you to determine the initial velocity rate, that v sub 0, with increasing substrate concentrations, and it gives you a curve basically like this. And so when you do experiments like this, where you're plotting basically the initial velocity and the substrate concentration, this relationship can be described by an equation called the Michaelis-Menten equation. So just to reorient ourselves, so we have an equation where enzyme plus substrate is converted to an enzyme substrate complex in a reversible reaction with K1 as the forward rate constant and K -1 as the reverse rate constant. And then that enzyme substrate complex is going to get converted into enzyme plus product with a rate constant of K2. So the Michaelis-Menten equation explains the relationship between the initial velocity and this increasing substrate and just the substrate concentration of the reaction. And so the Michaelis-Menten equation is v sub 0, so the initial reaction rate, equals the v max, which is the maximum velocity of the reaction, so the point at which it plateaus and the enzyme can't convert substrate to product any faster, multiplied by the substrate concentration over the substrate concentration plus Km, which is called the Michaelis constant, and we'll talk about this a bit more shortly. So this is the Michaelis-Menten equation, and it allows us, as I said, to understand the relationship between the initial velocity of an enzyme catalyzed reaction and the substrate concentration in that same reaction. So the Km is the Michaelis constant. And so the equation for that, so how we derived Km is basically taking into consideration those different rate constants all together. So Km equals K to the -1 plus K2 over K1. So it's basically the conversion of enzyme substrate either back to enzymes plus substrate or forward to enzyme plus product over the conversion of enzyme plus substrate into that enzyme substrate complex. So don't get too lost in the details here, but basically the Michaelis constant is describing the properties of the enzyme substrate interaction. So how quickly that enzyme substrate complex is able to form, how quickly that enzyme plus substrate is able to form the enzyme substrate complex, and then how quickly the enzyme substrate complex either converts to enzyme plus product or converts back into enzyme plus substrate. So the Michaelis constant is just a constant accounting for the properties of the enzyme substrate interaction. And then V max, which is the other new term in the Michaelis-Menten equation, is the maximum velocity. And it can be described by the equation Vmax equals K2. So that's the conversion of the enzyme substrate complex into enzyme plus product times the total enzyme concentration. And so that'll be known in these experiments because you're putting in a set amount of a set concentration of the actual enzyme. So this describes the velocity of the reaction when all of the enzymes, so the total enzyme concentration, is bound to the substrate. So that's as fast as the reaction can possibly go. So if you have, X moles of enzyme, and then all of those enzymes are bound to substrate, they're all converting from that enzyme substrate complex to enzyme plus product. That's as fast as this relation that the reaction can go. So the Vmax is the maximum velocity. There's when all the enzyme is bound to substrate, the reaction cannot go any faster than that. So this is the Michaelis-Menten equation, and it's a very useful equation for understanding the relationship between a reaction rate and the substrate concentration, and it gives us these two additional variables. The Michaelis constant, which describes that relationship between the enzyme-substrate interaction, and then Vmax, which is the maximum velocity that an enzyme can turn a substrate into product. So because Vmax and Km are such important variables in enzyme kinetics, I'll go over them a little bit more, even though I'm going to repeat myself a little bit. So first we'll talk about maximum velocity. So in this graph that we're showing here, you can see that the initial velocity is determined, again, with a fixed amount of enzyme and increasing substrate concentration. So the maximum velocity, Vmax, is described by the equation of Vmax equals K2 times the enzyme total. And of course, remember that K2 is the conversion of the enzyme substrate complex into enzyme plus product. And ET is the total enzyme concentration in the experiment. So the Vmax, therefore, describes the velocity of the reaction when all the enzyme is bound to substrate. So adding more substrate to the reaction will not increase the rate. So if you have this experiment, you have a fixed amount of enzyme, which would be concentration of E, the total concentration of E, and you keep adding substrate. At a certain point, you're going to add enough substrate where all the substrate is going to be bound to enzyme, and the reaction really can't go any faster. And that's why you get this plateau effect where the reaction is going to slowly, reach asymptotically to this level, which would be the Vmax. So the Vmax is shown right here, and this would be this line that's as fast as the enzyme can basically convert substrate into product. And so now the Michaelis constant, an interesting thing about it is the Michaelis constant is the substrate concentration at which the velocity is 1/2 of Vmax. So if we take our Michaelis-Menten equation here, so V sub 0 equals Vmax times the substrate concentration over the substrate concentration plus Km. So if we think about the actual substrate, so Km is an actual substrate concentration. So if we look at Vmax equals the substrate concentration over the substrate concentration of the experiment, and then we add in that substrate concentration of Vmax, what happens is this basically turns into Vmax over 2. So the Michaelis constant is going to be the substrate concentration at which the velocity is 1/2 of Vmax. And so you can see that over here on this equation. So we have Vmax up here. And then if you take 1/2 of the maximum velocity, remember here this y-axis is the velocity of the reaction, and then you take it down, The x-axis is the substrate concentration. So the Km is going to be that substrate concentration at which the reaction is half of the maximal rate of the total of the V max, of the maximum velocity of the reaction. So this describes how the Michaelis constant describes how the enzyme and substrate interact. And so it's unique for each enzyme substrate interaction. So it's a very good way to describe the actual kinetics of a specific enzyme substrate interaction. So remember, Km can also be described as K to the negative 1 plus K to the 2. So these are the conversions of the enzyme substrate complex back to enzyme plus substrate or forward to enzyme plus product. And that's over K1, which is the formation of that enzyme substrate complex. Now, one of the very useful features of Km, because it describes this enzyme substrate interaction and because it describes the substrate concentration at which the Vmax is, or the velocity is half its maximum value. So the lower the value of Km, the stronger the enzyme substrate interaction is going to be. And the higher the value of Km, the weaker the enzyme substrate interaction has to be. So take some time and think about that concept. It's very important, and Km is a very important variable for understanding how strongly an enzyme interacts with a substrate. But if you have a low value of Km, where basically you don't need much substrate to get to half that maximum value, that means that the enzyme has a very strong affinity and a very strong interaction for the substrate. If your Km value is very high, then that's basically what that's telling you, is that you need a large amount of substrate to get to half that maximum velocity. You need to keep adding more and more substrates. And that's because that describes a weaker interaction between that enzyme and the substrate. So that's Km. It's a very important concept. It's very important for you to understand both that Km is the substrate concentration at which at which the velocity is half of Vmax, and that the lower the value of Km, the stronger the enzyme substrate interaction, and the higher the value of the Km, the weaker the enzyme substrate interaction is. So here your book is giving you some different Km values or Michaelis constant values for some different enzyme interactions with specific substrates. And so we can already determine, looking at this graph right here, we can determine which enzyme substrate combination has the strongest interaction. So remember that the lower the Km, the stronger that interaction is going to be. So in these enzymes, lysozyme interaction with hexa-N-acetylglucosamine, it has the lowest Km of only 6 micromolar. And so this reaction, for that interaction between lysozyme and hexa-N-acetylglucosamine, you only need 6 micromolar of substrate for the reaction to get to half its maximal velocity. Therefore, the weakest interaction in this table here would actually be carbonic anhydrase, its interaction with CO2. So that has a Km of 8,000 micromolar. So you need a lot more of this particular substrate for the reaction to get to half its maximum value. And so it also turns out that the Km for most enzymes, substrate interactions that are important for biochemistry, are typically just above the physiological substrate concentration. So you can see here in this graph, here's half of Vmax, and then here's the Km, and then this pink region here is showing the physiological concentration of that particular substrate. So this allows the enzyme to be more sensitive to small changes in substrate concentration. So if you go from here to here, then that's quite a big difference in the velocity of the reaction. So if the Km was much lower, let's say the Km was down here, then this would basically be plateaued, and those small changes in physiological substrate concentration, the enzyme wouldn't be able to sort of respond or change its reaction velocity in response to that. So it's an important concept. So the Km for enzymes are typically just above the physiological substrate concentration, and this allows the enzyme rate to change in response to small changes in substrate concentration. So here's a practice problem we can go through. So this is going to be, probably pretty similar to something you might see on the exam, just to make sure that you understand the concepts of Vmax and Km and initial reaction velocity and all of that stuff. So in this question, it says enzyme A and enzyme B use the same substrate. And from this graph below, which enzyme has the lower Km and therefore the higher affinity for the substrate? Now, from this graph here, we can already sort of estimate. We could see what Vmax is. So, of course, Km is the substrate concentration at half of Vmax. So this would be half the maximum velocity at this point right here. So if we go over for enzyme A, the substrate concentration at half Vmax would be right here. And for enzyme B, it would be right here. So this is showing us that actually enzyme A has the lower Km for that particular substrate, and therefore it has a higher affinity for the substrate. Enzyme B has a higher Km, so it would have a lower affinity for the substrate. It needs more substrate to get to that to that half that maximum velocity. Now, another variable that's very useful when thinking about enzyme kinetics is turnover number, or what's called kcat. So this is the catalytic rate constant, or also commonly called the turnover number. So the turnover number describes how quickly an enzyme can convert substrate into product. So it's really the number of substrate molecules that can be converted into product per second. So the higher the turnover number, the faster an enzyme is going to be able to convert that substrate into product. Now, turnover number, you've actually already seen the variable for this, so it's just K2 from this equation. Enzyme plus substrate is converted into enzyme substrate complex. K1 is the forward rate constant. K negative 1 is the reverse rate constant. And then K2 is the conversion of enzyme substrate to enzyme plus product. So K2 describes how quickly the enzyme substrate complex is going to turn the product, can be converted into enzyme plus product. So the turnover number K2 is also sometimes called kcat, just because it describes that catalytic turnover constant. Now, this also gives us some other information. So we've also seen K2 in the equation for Vmax. So if the enzyme concentration is known, so the total enzyme concentration, then kcat can also be described by Vmax equals K2 or kcat times the total enzyme concentration. So again, this makes sense. So Vmax is the maximum velocity. How quickly is that total amount of enzyme going to convert the substrate into product? And so we can also write this equation as Vmax equals kcat times the total enzyme concentration. And so kcat would equal Vmax over the total enzyme concentration. So this equation here then describes exactly what I told you turnover number was. It describes the number of substrate molecules an enzyme can convert to substrate when the enzyme is fully saturated with substrate, when it's at its maximum velocity. So now we see this table here. You can see different turnover numbers for different enzymes. and carbonic anhydrase is on top. So I mentioned in a previous lecture, anhydrase is one of the fastest enzymes known, converting CO2 to H2CO3, and its turnover number, so its kcat, is 600,000 molecules per second. And so, yeah, so turnover number is a very important um, variable in enzyme kinetics, and it describes how quickly an enzyme can convert substrate into product. So the number of substrate molecules converted into product per second. So now that we understand how kcat works, we have a new variable. We can use that to get a new way to look at enzyme function or enzyme rates called catalytic efficiency, which is described by kcat over that Michaelis constant, Km. So in cells, the substrate concentration is usually much lower than the Km of an enzyme. And when that substrate concentration is much lower than Km, we can assume that all free enzyme equals the enzyme total, and we can derive a new equation from that. And so this equation would be V sub 0, so the initial reaction velocity equals the kcat over the Km times the substrate concentration times the total enzyme concentration. So when this substrate concentration is much lower than the Km, then the enzymatic velocity of the reaction is going to be based on all three of these variables. So kcat over Km, the substrate concentration, and the total amount of enzyme in the reaction. So kcat over Km is now the rate constant for this enzyme substrate interaction. And kcat over Km is also called the specificity constant. So it's actually, so that might have been a little bit confusing, but it's just important to understand that kcat over Km is a good measure of catalytic efficiency because it accounts for two different things now. It accounts for the catalytic rate of the enzyme, so kcat, the turnover number, so how fast an enzyme's converting substrate into product, and also it's accounting for that enzyme substrate interaction or affinity, which is described by Km. So we already sort of ran into this in those two tables that we looked at. An enzyme could interact strongly with a substrate, have a high affinity, but be very slow to catalyze that reaction. Lysozyme was an example for that. It had one of the lowest Kms, so the strongest affinity, but the lowest kcat of that reaction. So the high affinity, but low turnover number. Conversely, carbonic anhydrase had a low affinity for CO2, but it had a very high turnover number. So instead of using just kcat or Km to describe the kinetics of an enzyme substrate reaction, we have this catalytic efficiency variable that allows us to account for both turnover number and for the strength of that enzyme substrate interaction. So catalytic efficiency is a very important and very useful tool for describing enzymatic reactions. And so here we can see how useful that measure of kcat over Km, that catalytic efficiency, is to determine what substrate is ideal for a particular enzyme. So we have a table here that shows kcat over Km values for the protease chymotrypsin, which cleaves those peptide bonds in a polypeptide chain against different peptide substrates. So against glycine, the catalytic efficiency is quite low. It's a little higher for valine, and it's the highest for a substrate like phenylalanine. So this is showing us the ideal functional groups that act as a substrate for chymotrypsin for that peptidase activity. So phenylalanine, so a large aromatic amino acid, is going to have it's going to have the highest catalytic efficiency against that substrate compared to something like glycine or valine. So catalytic efficiency kcat over km is very useful for describing the interaction of a particular enzyme against different substrates. So now we're going to talk about the Lineweaver-Burke equation, which fixes some of the limitations of plotting out the Michaelis-Menten equation. So with the Michaelis-Menten equation, which you'll remember is VO, so the initial velocity equals V max times the substrate concentration over the substrate concentration plus Km. Now, when we graph this out, you can see if we run an experiment that Vmax value is going to be sort of approached but never fully obtained because you can't perfectly get it where all enzymes are 100% bound to substrate. You can get where 99.99, that, but you can't get an accurate number for it. So to get an accurate number, we use something called a double reciprocal Lineweaver-Burke plot. So we basically just take the reciprocal of this Michaelis-Menten equation. So instead of V sub 0, we have 1 over V0 equals Km over V max times 1 over the substrate plus 1 over V max. So this is a double reciprocal equation of that Michaelis-Menten equation. Now, the good thing about this equation is it gives us basically a slope. So 1 over V sub 0 would be y, and then Km over V max would be m, the slope. And then 1 over S is x axis. And then 1 over V max would be b. And so we get this y equals mx plus b equation, and that allows you to get a linear plot like this. And that way you could get actual accurate determination of both Vmax and Km. So if we look at this equation and this slope, you can see that the slope of the line equals Km plus over Vmax. And now the y-intercept equals 1 over Vmax. And then the x-intercept equals -1 over Km. So this is a very useful tool to figure out more precise measurements of Vmax and Km because you can determine them from the slope, the y-intercept, and the x-intercept when you plot 1 over the initial velocity over 1 over the substrate. So Lineweaver-Burke plot is very important for that, and also probably, it's easy to ask questions like this for the exam. As an example question, so just like with the Michaelis-Menten question I asked before, so enzyme A and enzyme B use the same substrate, and they have enzymatic activities described on this Lineweaver-Burke plot below. So from this information, which of the following are true? So enzyme A and B have the same Km and Vmax values. Do they have different Km and Vmax values? Do they have the same Km and different Vmax values, or different Km but the same Vmax values? So we should be able to determine this from this Lineweaver-Burke plot below. So we know the slope of the line is Km over Vmax. So and the slopes of the lines are different, so we know at least one of those variables has to be different. If we look at the x-intercept, which describes 1 over Km, we can see that these are the same. So enzyme B and enzyme A have the same Km value for both of them. But however, they are different on the y-axis, or the y-intercept, and the y-intercept describes 1 over V max. So in this particular example, enzyme A and enzyme B have the same Km value, but they have different V max values. So, and the answer B would be correct. And so now that we've gone through all these kinetic parameters and everything, we'll end the lecture today by talking about how to describe different biochemical reactions that have multiple substrates. And so multiple substrate reactions are basically divided into 2 groups. So one are sequential reactions, which is shown here, which occurs where all the substrates have to bind the enzyme before any product is released. So a complex of the enzyme with both of the substrates is formed. So in this example, we have the enzyme. It has to bind both NADH and pyruvate before it can convert into the products of lactate and NAD+. Now, the different type of reaction from that would be what's called a double displacement or ping pong reaction. And this happens where one or more of the products are released before all the substrates bind. So a modified enzyme intermediate will form in this type of reaction. So you have your enzyme here, and then it binds aspartate. So now you have an enzyme aspartate complex, and this will be converted, and a product will be released, oxaloacetate. But the enzyme is actually modified by this. So there's a modified enzyme intermediate, and that modified enzyme intermediate is now able to bind a different substrate, alpha-ketoglutarate, and then alpha-ketoglutarate, the product will be converted, and the product glutamate will be released, and then you have the enzyme as it was in the beginning. So this is called a double displacement reaction, also called a ping pong reaction, because substrates and products are basically bouncing off the enzyme. You have aspartate, and then bouncing off, turning into oxaloacetate, and then after that you have alpha-ketoglutarate, and then bouncing off, converting into glutamate. So sequential reactions, this is where all the substrates bind and then you release products, or a double displacement reaction where one substrate binds, releases a product, but you have a modified enzyme intermediate that can then bind a second substrate, releasing that product. So these are 2 different groups of biochemical reactions that have multiple substrates. All right, so you've made it through enzyme kinetics, and I hope you all have a much better understanding on how scientists measure the activity of enzyme catalyzed reactions. So we went over several variables associated with enzyme kinetics, and you're gonna need to understand all of these different concepts for the exam. So you need to know that enzyme rates are measured using a constant concentration of the enzyme, and then the initial velocity, so V sub 0, is measured against an increasing substrate concentration. And we also went over the Michaelis-Menten equation, which introduced things like Vmax, which is the maximum velocity of a reaction. So it's the point at which the enzyme is completely saturated with substrate, so it really can't go any faster. And we also learned about the variable of Km, the Michaelis constant, which describes the substrate enzyme interaction. So you need to remember that the lower the Km, the higher the affinity the enzyme has for the substrate, and the higher the Km, the lower the affinity the enzyme has for the substrate. We also went over turnover number, which is Kcat, which is how quickly an enzyme can turn over the substrate, so how quickly it can convert substrate to product. And we went over catalytic efficiency, which is Kat over Km, which takes into consideration both that turnover number and the affinity of the enzyme for the substrate. And lastly, you're going to need to know how to interpret Lineweaver-Burk plots, because these allow you to get more accurate measurements of both Vmax and Km. And you need to know the difference between a sequential and a double displacement or ping pong reaction. So I hope you enjoyed the lecture. Please let me know if you have any questions.